Is Claudia on already? Or?
She's on, yes.
Okay, hi Claudia. Hola, bienvenido. I hope I got that right.
Hola, gracias.
Practicing. Yeah, I've been practicing. So welcome to Arturo Lopez Pineda's doctoral dissertation. Big welcome, everybody. And every PhD student has a unique journey towards their defense and to their ultimate doctoral dissertation that they hand off to their committee. And I think Arturo has had his own very interesting, exciting personal journey. I think it began in, what is the year it began? Like 2010. And he's been in the Department of Biomedical Informatics since then. And he actually came to me in the probe lab and started his work in 2012. So it's been a very short journey, but a very exciting one for him over the last couple of years as we sped along to do this dissertation. And I think it's a lot of credit goes to his hard work and perseverance to have managed to finish so much in such a short time. So without further ado, I'd like to turn the stage on to Arturo, and he's going to present his doctoral dissertation entitled Bayesian Frameworks for Parsimonious Modeling of Molecular Cancer Data. Thank you. And before I forget, I should introduce his esteemed committee, starting with Professor Cooper and Professor Vishweshwaran, whom we all know, and Professor Claudia Rangel, who is from Mexico.
Right.
So welcome all of you. Yeah, welcome and thank you very much. He has 45 minutes to present his defense, and after that we'll have public session for 15 minutes, and then followed by a private session with the committee.
Thank you very much, Banati, for the introduction, and everybody for being here today. As Banati mentioned, I will be talking about parsimonious framework for cancer data. So the picture, the introduction is a nice butterfly, but I'm sure you all know this particular species, which is the monarch butterfly. It's a very important insect for North America because it travels from Canada to Mexico every year and goes back every year. So the question is, how does this insect start its journey? And so the people living in Mexico, they believe that this insect was their ancestors, and so they only knew that this insect came to them every year. But their answer to this journey was, it's my ancestors, and so they didn't know the other explanation existed. So it wasn't until very recently that some scientists decided to track this insect, and they discovered a different explanation. So the different explanation is that this insect is following the magnetic fields of the Earth. So there are two explanations here for the same phenomenon. So which one do we trust the most, right? Only one insect or millions of insects, but they all have the same two-way approach. So which of these two approaches is it the one that we're going to choose? And so there is this idea of parsimony, and that's where I want to start. The parsimony comes from the Occam's razor methodology, which tells us that the simplest explanation is the most probable explanation. So how does that translate into our field of machine learning? Well, parsimony tells us in machine learning that between two models that explain the data well, we would like to take the model that has the fewer amount of variables or the fewer amount of parameters to predict new data. And why we want to do this is because by parsimony, we can obtain models that are still quite good in terms of classification, but we are avoiding overfitting for future data. And that's the concept where I would like to start with parsimony. And I will develop some of these ideas further in the presentation. So what is the problem that we're trying to address here today? Well, I would like to talk about the burden of cancer and how cancer in the world, both in developing countries and developed countries like the US, it's a global disease that affects millions of people, and these are just rough numbers and estimates from the WHO. And you can see that for both male and female, the numbers are quite high in terms of estimates of new cases and, of course, also estimates in terms of deaths. And particularly, my focus of my work was on lung cancer, which is the most prevalent in male, and breast cancer, which is the most prevalent in female. But we also cover a little bit of the others, and we explore some of these data sets from the Cancer Genome Atlas, which is a publicly available data set resource in the United States. So this is a good motivation of why to do research, and a possible why to do research in cancer. And then a possible path for research is genomics. And in genomics, we have different ways of approaching this type of data. But this is just a schematic of a distilled model of how research works in genomics. And it's, of course, there are multiple frameworks that can be available, but this is just like a summary of them. So you have a set of patients that are extracted, from whom they are extracted a biopsy, and then this biopsy is processed and analyzed with genomics. And we would like to have a modeling. And this particular blue box is where I will be developing my frameworks. So what is the problem? In the pre-processing and design steps, there are a lot of algorithms already there existing, and in the functional analysis and validation of findings, it's a big step to go from purely data to the validation of findings. And what do I mean by that is to take a few genes that can describe a specific phenomenon in cancer. For example, to tell us the difference between subtypes of cancer, or the difference between different tissues in cancer, or so. So, but the thing is that I'm going to be focusing on modeling these types of cancer data. So let's start with some complex genomic models and drill down to something simpler. For example, here in Nature Review in 2001, the need for a networking approach was already recognized. And specifically because they were saying the amount of data from genomics is too high and the dimension is too high, that we need to have some networking approach to help us understand better. And so there are some examples of networks that have been developed over time, and these are fairly recent examples. But you can see that these networks and models to describe different types of cancer data, they have around 30 to 50 or maybe 20 number of nodes, which each one represents a gene. But still, it's not very easy. For example, in the one in the middle, it's not very easy to see from a human perspective which is the most important gene, or the subset of genes that we would like to focus on for human experimentation. It's very important for computer purposes, but not for humans. And some other models are also in the form of machine learning models. And this is just an example from Alferis et al. In 2002, where they developed a set of different classification models, support vector machines and neural networks with different parameters, and they basically had a very good accuracy, around 50, 60s, maybe 80% of accuracy in these classification tasks. Now, the number of genes is around 388. So they kept exploring. You can see it's still not perfect, and they kept exploring, and the same group now, where the first author is Statnikov et al. But the first group developed a new set of algorithms, more modern ones, and their accuracies went from 84 to 94, or even 1, very close to 1. But these are the number of feature operational units, which is the same as features in their case. So they went from 308 to have some algorithms that are around 8, 51, 20, but it's not really consistent across all the different data sets. So each row represents a data set, and each column represents a machine learning model. So SVM with feature, I forgot what they are, but SVMs, and some other refinements of their algorithms. So in this paper, they are recognizing that the main focus was to improve the classification accuracy, not so much on the selection of operational units or features. And they do recognize that there is a need for having models that can capture the same complexity with fewer number of variables. And so this is the motivation of my work. And so to keep exploring this, we had some methodology that originally published in March this year in the EMEA TBI conference, and where we were very happy to win the first prize awards with Vanity. So the original publication is this. So we start with some data from the Cancer Genome Atlas, and we filter this through two methods: discretization using Fayyad-Irani MDLPC, minimum description length principal component, principal cut, and then we select the best features with the Relief F algorithm, which is also very well known in the machine learning literature. Then we select the best features and build a Bayesian network model with using Dr. Cooper's EBMC algorithm, which is also state of the art. And from this, we would like to extract a junction tree to select some new knowledge that can be interpreted by the experimental researcher. So this new knowledge, it was in the form of a subset of features and maybe some structure, but we weren't really clear of what is the new knowledge that we would like to have at that point. So we kind of updated our framework. You can see that our framework is called the Junction to Knowledge Framework, J2K. And now we know that we would like to extract a subset of these features. So the new knowledge is in the form of a subset, which is a parsimonious model. And this would provide us with a clique or a small number of features that are interconnected. And this is what we're going to provide to the experimental researcher. Okay, so the first part is the classification section. And the second part, which is the junction, which we already classified with the machine learning. And then we would like to extract some features from the model, so for the full model. So this is why we call our framework the post-classification framework for knowledge extraction. And this is what we are hoping to lead to new knowledge. And let me give you some examples, an example of how we approach this so that we can create new knowledge in breast cancer. But before that, let me talk about the thesis. So the thesis is that the J2K framework, which I just showed you, yields parsimonious computational models with two properties: high classification accuracy, and it's going to be evaluated using area under the curve, and also a small number of variables. And by small, I mean around 10. It's very debatable because it's human purpose. How many is few? But for my purposes, I was thinking around 10, seven number of features. There's a reference about how many pieces of information a human being can handle, and it's around 7 plus minus 2. So 7 plus is 9, and minus 2 is 5. So around that is going to be the small number of features. So this is my example. Some of you might recognize yourselves in this picture. So from a set of possibilities, I would like to choose one, right? And so I just need to show you one example of this. So think about breast cancer. We have a set of data set here. Each row represents a sample from a patient, a biopsy, and each column represents a gene in case of the gene expression, and or a methylation site in case of the pink section here. So the TCGA is the first element in this code, right? So it's saying it's coming from a TCGA data. It's BH, which means it's coming from Pittsburgh. In this case, this is a sample from Pittsburgh. I don't really remember if it's BH Pittsburgh or not, but let's for now think that it's Pittsburgh. Then this is the ID of the patient. So a patient can have multiple samples. For example, in case of breast cancer, it can be the tumor sample, or it can be the adjacent sample, or it can be the other breast. And this number here is indicating if it's a tumor sample or a normal sample. And A just represents if it's the first sample that it was taken, or it's the replicate. In case of the replicates, it can be B or C or D. You can increase it. I'm sorry. The remaining are the expression of each gene. So, for example, ELMO2, it has an expression of 0.87, as accounted already by normalization, and this is the data that was corrected for batch effect and quality control from the TCGA data, so we can trust that this data is coming from reliable sources. That's very important in genomic analysis, that whatever we do in machine learning, we are still dependent upon the quality of the data that we're given. And the remaining section is the class. In this case, this patient has a stage 0 or 1, as defined by the TNM section. And then the others are 2 to 4, which means they are advanced patients. And the same for, this is the same patient. You can see the same patient in the gene expression and the DNA methylation. I'm just going to be showing both data sets at the same time, and I will tell you why later. The first step in our J2K process is to discretize. So we go from 0.87 to bin that can go from, in the case of BDCL3, from 0.97 to infinity, and from minus infinity to 0.97 with a closed parenthesis. So you can see that for this gene, there are two bins, two distinct genes, that one is saying this is high expression and the other one is low expression. Of course, we are losing information here because we are going from a continuous value to a discrete value, but it has been shown in the literature of machine learning that this process improves classification performance. And the other advantage of doing this is that most of the several algorithms in machine learning also require the use of discrete variables. So one of the limitations is that, of course, we are going to discrete area, but we are still obtaining good results, so we're okay. And the same goes for methylation. From there, we select the best features. And by the best features, I mean the best n number of features, and we can debate on how many are the best and sufficient. So we selected 50, and I'm just showing the best 15 here, but we selected 50, 50. Why 50? It's because the number of features that we are selecting depends upon the scoring that you are, or the expression that you would like to have, right? So in this case, we're using relief F, and you can see the relief score right next to each gene. And for example, the best gene here is LCE3B, which has a relief F score of 0.14. And so we keep it going, and the best 50 genes are the best ones that can classify it. So there are some references in the literature of how much is enough to have, right? 100, 200, 1,000 genes. And there is a reference of having a number between 30 and 50 that is just enough for capturing the complexity of the model. This is of course experimental and hasn't been sufficiently proven, but for the purpose of this talk, we are going to trust that it's a good resource. Then we create a machine learning classification model, in this case a Bayesian network using the EBMC algorithm. And it creates two models, the gene expression model and the methylation model. From these models, we can see that the number of variables is already reduced, and the classification of this model, the classification accuracy of this model is very high. So, but the still gene expression model is a little bit too crowded, right? So we kept going, and then we built a junction tree, and the junction tree is coming from this directed structure that has direct connections from the genes from the class to the genes to an undirected structure. So the undirected structure looks like this. These are the junction trees. What we do to transform from a directed to an undirected is to first moralize the graph, which means connect every node that has common children. They are going to be connected, and then remove directionality, triangulate, and then a series of steps until we arrive to the junction trees. Every circle here represents a junction. Represents a clique, which means a set of genes that are highly connected in the undirected structure, which means everything is connected to each other. And every square represents a junction, or the connection, the common genes that are, the things that are common between the junctions. So for example, in the gene expression model, there is only one junction, which is the class. So only the class is common to every other clique. And in the case of the DNA methylation, there is a junction there that can have a few other genes there. So these are the junction trees, and from here we would like to evaluate the best clique, the best possible clique. So how do we do that? We go back to the gene expression Bayesian network, and then we select the cliques from, let's say, clique number 1. We select the genes from there, which are these ones. And so the expectation is to see if we remove the rest, how well does this new network will classify. So we do exactly that. We remove the rest, and we have a new network that is parsimonious. So remember that this click is coming from a highly connected network. You can see that there are some arrows that are missing. For example, AQP4 is missing an arrow for OR2W3, right? But it was connected in the undirected structure. So in the undirected structure of the junction tree, it's selecting this click. So then we evaluate every single one of them. And we see, okay, for methylation, we have a complete network, which has a classification performance of 0.72. And in the gene expression, the complete network has a classification performance of 0.53. For every click in methylation, the area under the curve is smaller than the complete network. But when we make a statistical comparison, we can see that only click number 5 is not statistically significant, but it's not significantly different, which means it's equivalent to the complete network. And we can substitute click number 5 for the complete network. Okay. Similarly, for gene expression, we can see that every click is better than the complete network. So now the statistical comparison has to be the opposite. Now we would like to see that they are statistically significantly different. And so every click is statistically different from the complete network, but differently better. So in case you see p-values in one section and p-values in the other, they have different objectives. In the gene expression, because they are better, you have to take a look into area under the curve and also p-values because it's a double-sided. So we like to select the best ones in this, right? The best ones that have less variables. And the number of variables in the methylation is coming from 12 to 5, and in the gene expression from 25 to 9. And remember that I told you that it was between 7 plus minus 2, so still around the boundaries. And we select these two. So the complete network are okay, but then we were wondering, okay, we have data from the same patients for methylation and gene expression. Can we do better than just using one single gene? And then we come up with this other framework that we call MODI, which is multi-omic data integration, also known as President of India or Prime Minister. And we combine these two networks by using a latent variable. So the original class becomes a latent variable, and we create another class that has the same two states. Remember, early stage versus late stage. So these are the complete networks, and we have 3 arrows there, A, B, and C, that can be explored. And we have that, we have explored that. But for now, let's say that we want to use all 3, because we know that methylation has some effect in gene expression, and the gene expression will have some effect in the progression of cancer. That's our hypothesis in this model. So that's why we selected A, B, and C in this case. But what happens if we use the parsimonious model? So this is already parsimonious, but this is even more parsimonious, right? We are selecting click number 8 and click number 5 from both gene expression and methylation. And we have a smaller model that is multi-omic. At the same time, it's providing us with a lot of information for the gene expression and methylation of breast cancer progression. So I would like to point out the gene RRM2, double RM2. This gene was not evident from our previous models. Let me just go back quickly, and let's try to locate it very quickly. It's in the bottom of the gene expression. Can you see it? Maybe a little bit. It's right on the gene expression section. It's in the bottom. It's not very evident that this is an important gene from this network. So, but here, why is it evident that it's an important gene? Because it's the only gene that it's not directly a children of the class. And why is that important? Because it's regulating the other genes as well, in conjunction with the phenotype. So it's giving us a little bit of more information on where to look. And that's what experimental validation would like to do. We would like to see, these are the genes that we would like to see further and explore in the lab. So what happened with RRM2? And when we look in the literature, what's going on with this gene, we find more than 79 publications. And the most important things about gene expression in this particular gene is that it's directly related to breast cancer progression. You can see, for example, the first one is RRM2 is estrogen negative breast cancers, prognostic therapeutic significance, and then prognostic marker for breast cancer association for poor survival in tamoxifen resistance. So what is this paper? What are these papers telling us? That RRM2 is directly related to breast cancer, but not only that, it's directly related to breast cancer progression, which was the original task where we started from in the breast cancer classification before. So we can do the same thing for the rest of the genes in the model, which we have done. And if you want, I can talk to you later about these other genes, which are all related to breast cancer. So, and that concludes the gene expression J2K example. But we wanted to know, okay, what is the contribution of each component from the J2K to the parsimony of the models? Because my thesis is that it's contributing to create parsimonious models. So instead of having just one example, we had now 4 different data sets: lung cancer, lung adenocarcinoma, lung squamous carcinoma, breast invasive carcinoma, ovarian carcinoma, and colon cancer. And we have, of course, gene expression, we have methylation, and we have different classification tasks: tumor versus normal, subtypings, adeno versus squamous. We have staging, early stage versus late stage, and different cases, number of imbalances between these. But most of them are really well balanced, except for number I, which is extremely imbalanced. And ovarian cancer, it's very few normals, so it's hard to get. But just keep in mind these numbers, and there are some handouts in the table if you want to keep looking at them. So the evaluation of this model, the J2K model, comes with selecting different algorithms, because so far I've been using the same ones over and over. And so we wanted to know, if we change one algorithm here, what's going to be the effect in classification performance and the number of variables that we get. So we have different variables for each one of these elements. Okay, so the first is that instead of using Fayyad-Irani discretization method, we also use Kononenko discretization method. Both of them are minimum description length algorithms. It's just that the criteria that they are using for selecting the best cut is different. So Fayyad-Irani uses information gain, and Kononenko uses kind of a relief F criteria where they select the best nearest neighbors. So for every data set, they reduce the number of variables. In the case of methylation, you can see the variables that have the data sets that have M, those are methylations, and the data sets that have a G, those are gene expressions. So the gene expressions go from 17,000 to around 2,000, 1,100, and the methylations comes from 27,000 to around half, a third, depending on the variable. But the classification performance, so we discretize and we obtain 10,000 variables, for example, in number 8. And using these 10,000 variables, we create a naive base and classify using a 10-fold cross-validation performance. And we can see that we get an awesome performance, right? It's 1.0, very close to 1. So it's not really 1, but because these are rounded numbers, it actually says 1, but it's very close to 1. So it's okay, we have good classification performance, but we're missing the part of having a small number of variables. So far, we know that the difference between Fayyad-Irani and Kononenko, they look similar, so statistically indistinguishable, and we cannot really choose between one of them, and so this is, I guess, the component in J2K is saying discretized. It's not really telling you which algorithm, so we can use either Kononenko or Fayyad-Irani. But so far, we are going to choose one, which is Fayyad-Irani. It's very well known. So the next step is feature selection. And here we have a relief F, we have information gain, which are also very well-known algorithms in machine learning. So for relief F, we are selecting the best 50, and for information gain, the same, the best 50, and then we create. So we discretize first MDLPC, that's Fayyad-Irani, then relief F for information gain, and then an naive base with these 50 best features. So the numbers improve from naive base, and the numbers are not very different from each other in terms of classification performance. And we can say here that feature selection improves significantly the classification performance compared to not using feature selection. And also that between information gain and relief F, there's no real clear distinction on which one to use, so they perform similarly. But we are using relief F just because it's also very well known and it has been proven to have a good significance. So the next part is using Lima and relief F. Lima is another feature selection algorithm that is very, very well known in the bioinformatics community, which basically helps researchers find differentially expressed and differentially methylated genes. But Lima starts with continuous values. And here we are testing two things. We're testing whether we should use Lima instead of relief F, or whether we should use a discretization and then feature selection mechanism, or feature selection and then discretization mechanism. So the number of variables in Lima is significantly reduced, but the classification performance is also reduced. And why is it so that it's reduced? Because we select Lima, the best 50, first, and then MDLPC also removes some of these variables. Why is it removing them? Because when you have a variable without cut points, then MDLPC will take it out because it's not relevant for classification. And it doesn't happen, the same doesn't happen with relief F. So relief F maintains the same 50 after you discretize. So we can extract from here that relief F significantly improves the classification compared to Lima. But it's preferable to discretize before feature selection. And I'm not saying this is a very hard statement, it's just saying that it's preferable. Because depending on the feature selection method that you use, it can change a little bit. So in the case of Lima, it's of course better to use relief F. But, and we can talk more about this, of course, because we are writing a paper about this particular comparison between relief F and Lima. Okay, so now we have a set of features that are highly good at classification. Then we want to build a model, and we were using EBMC, but there are other models that also provide a connection, a classification Bayesian classifier. For example, TAN is one of those classifiers, and naive Bayes, which is the one that we have been using so far. The problem with TAN and naive Bayes is that it doesn't reduce the number of features, and EBMC is on top of the feature selection that we were using. EBMC is reducing it even further. So from the 50 original variables that were selected from the feature selection model, in TAN, we kept the same 50. We built a naive Bayes, and then the algorithm actually incorporates a tree structure underneath. So this is the tree augmented naive Bayes. It's a naive Bayes with a tree underneath. And in EBMC, it has a similar structure, but at the same time, it selects more efficiently the best variables. So EBMC and TAN here doesn't seem to have much difference in terms of classification performance, but it does have a lot of difference in terms of the number of features. And that's good. So in terms of classification performance, naive Bayes is of course better, and it has been shown that in the literature so many times, but not so much. I mean, it's significant, but it's not so much, and the number of variables is significantly reduced when we use EBMC. Comparison in terms of classification between EBMC and TAN is also not distinguishable in terms of classification performance, but it's distinguishable in terms of the number of variables. So in terms of parsimony, we would like to have EBMC, and that's the reason why we chose EBMC. And so we have models, and we would like to have a post-classification approach. And this post-classification approach comes in the form of selecting the best click. And for doing that, we build a junction tree. We extract every click, and then we evaluate all of them. And here I'm showing the best click, the one that was selected with the best classification performance. So this is, for most of these data sets, the numbers are terrible, right? Because it's 0.53. And for A and B, and for J, and L and K, the numbers are above 0.7, which is already okay. And the number of variables is reduced significantly for all of them. So we cannot really say that the junction is consistent. It's okay for some of these algorithms, but it's not consistent across all data sets. So we still have to do more exploration on why is that. So there is a second approach, which is from the EBMC search, we select the first iteration of the algorithm. And if we only select the first iteration and not, which is the iteration that has the best classification in terms of the way the search is doing, then we have only 10, 6, 3, around 10 variables with a very good classification accuracy, actually really good classification accuracy. There is only the case of H, of the data sets H and I, which you can see that none of these algorithms classify well. I'm sorry, J, H, and I. So J and H are the staging problems. So trying to classify a staging problem from primary tumor is a really hard problem. And the way we solved that was with the MODI framework, when we integrate both gene expression and methylation. Now, I is not a problem. It's not a very difficult problem, but the original data set was very skewed. And we don't have enough data. So we think that these numbers are okay because, the data set doesn't have the information that we want, or we would like to have more data to keep exploring these three particular classification tasks. So the first iteration of EBMC is statistically indistinguishable from the full EBMC. So what is actually quite an improvement, because EBMC has some parameters, but if we only want the first iteration, we don't have to provide all the parameters. We only have to say we want the first iteration, and EBMC will start. And that's we will roughly have to say how many variables is the top of our search. So in this case, 30. The first iteration of EBMC improves the classification compared to junction. But again, junction is still valuable, but we have to keep exploring, and it's not consistent, but it has some alternatives. Okay, so what is the conclusion? So the conclusion is, I will go back to my thesis, which was J2K yields parsimonious computational models. And if you remember, I said that it was two characteristics: high classification accuracy and a small number of features. So high classification accuracy goes for 88 on average, and 9 variables on average for the model that has MDLPC, Relief F, EBMC, and the first iteration of EBMC selected in a post-classification manner. And this is the average across all datasets. But of course, we have to drill down to every little dataset to find out the differences. So I hope to have convinced you that parsimony is important and it's feasible to do, and that's one of the main conclusions of this work. So future work for this is to investigate the structure of the junctions, because we believe that there is some functional meaning in the structure of the junction that we haven't explored, but for the sake of time, we decided to postpone it for another work. And so also explore novel strategies for Bayesian building, because we have seen that EBMC is very efficient, and that by refining a little bit onto the algorithm or selecting just when the first iteration, we can keep exploring parsimony. So that's another future work. Also expand the MODI framework, because so far I show you the integration between gene expression and methylation, but of course there are others like microRNAs and proteins and other type of genomic omics out there, so that we would like to expand that. And also, in terms of the data that we're using, we were using only tumor tissue from biopsies, but also perhaps use other types of tissues, other types of biopsies like liquid biopsy, which is a trending topic right now. So some of the contributions of my work is, of course, the J2K framework, which we presented in a paper, and the MODI framework, which is an oncoming paper. The development of EBMC for WECA, which is publicly available now, and novel findings in lung and breast cancer that we are in for example, they show the breast cancer RRM2 that I show you, and others that we have in our publications. And some of these publications include the Marco Ramoni Award. And here, Dr. said it's good that he's here because we just published a very good comparison of machine learning algorithms and other publications that are oncoming with in lung cancer in combination with the Pittsburgh Cancer Institute, and of course with Claudia in Mexico and the whole team of the BMI here, Jia, Henry, and Brian. So just to conclude, I just to show you some of my journey. As Panathi mentioned, it was quite a journey. So I've been quite around the US, right, and Mexico, of course. Most of this work was funded by the Fulbright Scholarship. And as I want to thank my parents for this presentation, and of course my committee members, Dr. Cooper, Dr. Bisbashbaran, my mentor, Dr. Krishna, and Claudia, which is in Mexico, and Dr. Rangel Escareño. Of course, Ms. Tony Porterfield, and here some of my professors, Rich Sui, which was my mentor in my master's project, Harry Hockheiser and Jerry Douglas, and the rest of the professors in the VMI and some of my fellow classmates, and most dearly to the Fulbright Scholarship, to the CONACYT Scholarship, and to all the funding agencies that supported my work throughout these years, and all the people, administratives that worked in these agencies. And I would like to take some questions now, and please let me know. Thank you.